Hello, Harper. Well, today we're going to be looking at biology. The atheist worldview is built on the idea that everything came from nothing, and within that everything, some chemicals self-organized and somehow became alive and began to reproduce. This is where biology begins, with reproduction. Though there have been many experiments over many years, there has been zero progress on finding out how the first living cell came to be. Charles Darwin had no idea where the first living cell came from, and he didn't worry about it. He just accepted that it came from somewhere and went on to develop a marvelous story about how every living thing, fish, birds, animals, flowers, trees, mosquitoes, and men, everything had descended from that first living cell. He described a process that he thought might produce this result and called it mutation with natural selection. The idea is that as an organism reproduces, there are occasionally small changes compared to the parent organism. And if those small changes caused the daughter organism to grow stronger than the parent, well, the daughter's children would become established in the ecosystem and would eventually overtake the original parent organism. Let's review a short clip from one of the videos we saw in the last letter, and that'll kind of refresh our memory on that.
Darwin's theory of biological evolution tells us that all life on Earth may have originated from a single, relatively simple reproducing creature living in the distant past. This idea is based on many observations, one of which is that when living things reproduce, children are often born with random new traits. Those with negative new traits are less likely to survive and reproduce. Those with positive new traits are more likely to survive and pass those traits on to their children. Over multiple generations, positive traits build up, allowing relatively simple life forms to evolve into complex life forms and even split into multiple different species.
So, Darwin saw small changes like these and concluded that millions of years of very small changes would add up to very big changes. And eventually, a fish would develop lungs and legs and begin walking on the land. Now, we'll focus more on these small changes a little later. This is Charles Darwin's general theory of evolution. Every living thing is descended from a common ancestor. The wide variety of life was brought about through mutations and natural selection, otherwise known as survival of the fittest. I want you to meet Dr. Michael Behe. Dr. Behe is a professor of biochemistry at Lehigh University. He holds a PhD in biochemistry from the University of Pennsylvania. He did postdoctoral work on DNA structure at the National Institutes of Health. And like every other biology major, he was taught that Darwinian evolution was a fact.
For the longest time, I believed that Darwinian evolution explains what we saw in biology. Not because I saw how it could actually explain it, but because I was told that it did explain it. And in schools, I was taught Darwinian biology. And through college and graduate school, I was in an atmosphere which just assumed that Darwinian evolution explained biology. And again, I didn't have any reason to doubt it. It wasn't until about 10 years or more ago that I read a book called Evolution, a Theory in Crisis by a geneticist by the name of Michael Denton, an Australian. And he put forward a lot of scientific arguments against Darwinian theory that I had never heard before, and the arguments seemed pretty convincing. And at that point I started to get a bit angry because I thought I was being led down the primrose path. Here were a number of very good arguments, and I had gone through a doctoral program in biochemistry, became a faculty member, and I had never even heard of these things. And so from that point on, I became very interested in the question of evolution, and and since have decided the Darwinian processes are not the whole explanation.
Darwin, in developing his theory, made 3 serious errors. Now, we've already discussed the first one, and we'll discuss the third one in a later letter. For now, we'll focus on the second one. Darwin thought the cell was a simple jelly-like substance. He called it protoplasm and assumed that it would be a simple thing to form from the right chemicals in the right soup. Now, to be fair, Darwin had no way to know what a cell really was or what it did. He had no tools that would allow him to look inside. Today, we know what's inside a cell, and it's exceedingly complex. There are 30 to 40 trillion cells in the human body. Every single one of them is a fully functioning, self-contained factory. Each cell has the systems and equipment in place to build new parts, move raw materials from one place to another, repair broken equipment, recycle broken parts that are no longer useful or functional, and replicate itself. It's an amazing world of activity in each and every cell. And every one of these little factories runs around the clock 24/7. So if you feel tired all the time, there's a good reason. You've got a lot going on inside. Well, all of these machines are made out of proteins. And Dr. Ken Dill is going to give us an introduction to proteins and how they function in the cell. He holds a PhD in biophysics and will explain more about this amazing machinery.
I would like to tell you about the protein molecule. Protein molecules are a class of chemicals in your body that do everything that run your system, your body, and other living systems. They make you walk and talk and think. They run your immune system. They run your brain. They run your heart. They digest your food for you. Now, protein molecules are the subject of Biology 101 courses everywhere. I am not going to tell you about that basic subject, but rather, instead, what I'm going to tell you about is 2 fairly new and very important developments focused on protein physics and protein engineering and the exciting implications they have for potential new technologies. First thing I want to do is just introduce you or remind you, for those of you who've seen this before, what a protein molecule is. They are very small. You have billions of trillions of them inside your body. And a metaphor you could say, if you could scale up a protein molecule to the size of a penny, the number of protein molecules you have in your body is the same as the number of pennies that would fill the Pacific Ocean. You have lots of them. They're extremely small. Now, on the other hand, having said this, let me say also that protein molecules among molecules are very large and big and complicated, and this is a picture of one. This is a picture of a relatively small protein molecule. The beads that you see on this diagram are atoms, and the little lines connecting them are chemical bonds. And a key feature of protein molecules is that they are long and stringy, and they are made of, I've colored here, these are false colors, chemicals don't have color. I've colored these beads along the chain. They look like beads on a necklace. The beads themselves are the components of protein molecules, and they're called amino acids. Amino acids are very simple chemicals. You can go down to your drugstore, you buy powders. They're a couple of bucks. They go by names like valine and isoleucine and glutamine and phenylalanine and things like that. Those are the component pieces. This is a picture of showing you the string. There we go. This is just a schematic representation that strips out some of the amino acids so that you can see 2 things. One is the basic long stringy character of a protein molecule, and secondly, what an amino acid looks like along the chain. I've stripped out all the rest. Now, the first major development I want to tell you about is a thing called the protein folding problem and the protein folding code. This is has been considered one of the grandest challenge problems for the last 50 years in biochemistry. The protein molecule has a code, and the question is how to break it. And what I want to report to you is that a community of biophysicists has come now very close, has solved many of these problems, both high-level problems and the detailed problems, and we can now compute the structures of proteins in computers. Here's what the code is. Think about this necklace, if you like, as being made up of pearls that have 20 different colors. The 20 different colors represent the 20 different types of amino acids, so they represent the different chemical types. I'm just using a metaphor here. Suppose you could string those pearls together in any order you wanted to: red, green, blue, blue, pink, and so forth. It turns out the way you string those pearls together, the amino acid sequence, determines how this long stringy molecule crunches down into a crumpled version, which we call the shape. Now, one string of colors, blue, blue, pink, will crunch into one shape, and that shape may become a part of your muscle. A different string of pearl colors will crunch down into a different shape, and that will be, let's say, an antibody protein in your body. So the code is how the string of amino acids encodes what the shape is. That's the protein folding problem. I want to describe some advances in that area first. And then the second part of the problem is, once you know the shape of the protein, how can you tell what it does and how it works? These are the two key components for understanding the functioning in your body of protein molecules. Now I'm going to show you the folding problem and what we're looking to try to do. This is a protein that's all stretched out. You see 4 red colored amino acids here. And the basic idea is this is a chain that needs to find its shape. And we know that these 4 red beads, we know after the fact that these 4 red beads need to find each other. It's very much like you walking through a crowd of people trying to find your close friends. It's a fairly random process, but sooner or later you can sometimes do it. Those red beads need to find each other. This is called folding. And if you look at the blue strands, what they need to do is they need to line up like train tracks. And what the gold part needs to do is coil up like a helix. And this is showing you the folding process. And one thing you'll notice is that these pieces form, and they do it systematically. The other thing you will notice, so this is the final shape, and this is the shape that your protein has when it's in your body. This is the shape it needs to function. But the other thing you notice about this movie is it's very jiggly. Why is that? It's because protein molecules are so small that they get banged around by water molecules. Think about riding a bicycle in a windstorm. You're trying to get somewhere, but you keep getting blown around. And that's how protein molecules have to function too. They have to deal with these little microscopic windstorms. So this is the shape of a folded protein. The folding problem and the folding code is the business about, if I knew what the sequence of amino acids is, how do I figure out what the structure is? Now this next picture is showing you a more realistic, bigger protein molecule. Most protein molecules are bigger than the one I just showed you. They often look something like this. And now I want to switch from talking about the folding problem per se to talking about mechanisms and functions. And the case I want to make for you is that proteins are machines. You have 20,000 odd different types of machines in your body, and then other kinds of living organisms have other kinds of protein machines. There's tens of thousands to hundreds of thousands of different machines. And the first case I want to make for you is that these are real machines. That's not a metaphor. They use energy, they spin around, they pump, they act to cause force and motion. This is just a side view, a static view of a thing that is a protein motor. That light gray at the top is your cell membrane. Above it is the outside of the cell, below it is the inside of the cell. And this is just showing you a static picture, and now what I'm going to do is to show you a dynamical movie of how this machine actually works. This is a picture of that This is a movie of that machine, and what you can see is it pumps acids, those are the little cubical things, in and out of your cell so that it keeps the pH balance in your cell. And the things down at the bottom, the little cards going in and out, that's where the energy comes and goes. And what you can see is a ribbon diagram. So that shows you the nature of the protein molecule. And a critical point that I want to make about that movie and about how proteins function in general is they function by shape and by shape changes. And you could see that protein molecule going back and forth as different chemicals come and go. Shape is the critical thing here. Next, I want to show you another type of rotary motor. It's one that does not pump acids in and out of cells, but rather it actually creates force. If you're a bacterial cell, you need to propel yourself around in water to find food. What you need is something like the propeller on the back of a boat. But life doesn't have propellers for various reasons. Instead, what it does is it has long tails called flagella. And so now I'm going to show you a movie of a rotary motor that runs the tails on bacteria. Here you see it. Those are the tails. Now you're zooming in, and you're going to see the rotary motor in a kind of a cartoony-like fashion. This, too, is a real protein, and it uses energy, and it creates motion. That's rotary proteins. Those are a couple of many rotary proteins. Now I'm going to show you another kind of protein. This is a motor that slides back and forth. It walks like 2 feet. It's like a drunken sailor. Some of you who are old enough may remember those old cartoons about Keep On Truckin', where you walk like this. That's what these things do. The ATP and the ADP at the bottom where the feet are, incidentally they're called heads, but where the feet are, that's where the energy comes in, and the motion happens in a general direction, but again, it's battered around by the water. And these kinds of motors are used in your muscle. So every time you do anything, it's sliding motions that are happening at the microscopic scale that cause that to happen. One of the great things about protein motors compared to real world, big world motors, if you like, is they don't make noise. And the last example I want to show you of a protein mechanism is this. Some proteins are valves and pumps. This is a case where this one too sits in a membrane, and it controls water flow in and out. Other such valves and pumps control lots of other things that go in and out of your muscles, and this is how your nervous system works, for example. If these kinds of proteins go bad, you get things like cystic fibrosis, for example. I've just shown you a few examples of tens of thousands of protein machines, but the case I want to make for you is just to convince you they are real machines, just at the super microscopic level.
So, proteins form real machines in the cells at the microscopic level. They burn energy, do real work, produce waste, break down, and wear out. Other machines either repair them when they break or take them apart to recycle the parts when they're worn out. Here's another video that goes into more detail about all these processes.
These are tiny molecular machines, and they are doing this inside your body right now. To understand why, we have to zoom out. Every day in an adult human body, 50 to 70 billion of your cells die. Either they're stressed or damaged or just old. But this is normal. In fact, it's called programmed cell death. But to make up for all these lost cells, right now, billions of your cells are dividing, essentially creating new cells. And that process of cell division, also called mitosis, well, it requires an army of tiny molecular machines. So let's take a closer look. DNA is a good place to start, the double helix molecule we always talk about. This is a scientifically accurate depiction of DNA created by Drew Barry at the Walter and Eliza Hall Institute of Medical Research. If you unwind the two strands, you can see that each has a sugar phosphate backbone connected to the sequence of nucleic acid base pairs, known by the letters A, T, G, and C. Now the strands run in opposite directions, which is important when you go to copy DNA. Copying DNA is one of the first steps in cell division. Here, the two strands of DNA are being unwound and separated by the tiny blue molecular machine called helicase. Helicase literally spins as fast as a jet engine. The strand of DNA on the right has its complementary strand assembled continuously, but the other strand is more complicated because it runs in the opposite direction. So it must be looped out with its complementary strand assembled in reverse, section by section. At the end of this process, you have two identical DNA molecules, each one a few centimeters long, but just a couple nanometers wide. So to prevent the DNA from becoming a tangled mess, it is wrapped around proteins called histones, forming a nucleosome. These nucleosomes are bundled together into a fiber known as chromatin, which is further looped and coiled to form a chromosome, one of the largest molecular structures in your body. You can actually see chromosomes under a microscope in dividing cells. Only then do they take on their characteristic shape. Otherwise, the DNA is more strewn inside the nucleus. The process of dividing a cell takes around an hour in mammals, so this footage is from a time-lapse. You can see how the chromosomes line up on the equator of the cell. Now, when everything is right, they are pulled apart into the two new daughter cells, each one containing an identical copy of DNA. Now, as simple as this looks, the process is incredibly complicated and requires even more fascinating molecular machines to accomplish it. So let's look at a single chromosome. One chromosome consists of two sausage-shaped chromatids, containing the identical copies of DNA made earlier. Each chromatid is attached to microtubule fibers, which guide and help align them in the correct position. The microtubules are connected to the chromatid at the kinetochore, here colored red. The kinetochore consists of hundreds of different proteins working together to achieve multiple objectives. In fact, it's one of the most sophisticated molecular mechanisms inside your body. The kinetochore is central to the successful separation of the chromatids. It creates a dynamic connection between the chromosome and the microtubules. For a reason no one's yet been able to figure out, the microtubules are constantly being built at one end and deconstructed at the other. While the chromosome is still getting ready, the kinetochore sends out a chemical stop signal to the rest of the cell, shown here by the red molecules, basically saying this chromosome is not yet ready to divide. The kinetochore also mechanically senses tension. When the tension is just right and the position and attachment are correct, all the proteins get ready, shown here by turning green. At this point, the stop signal broadcasting system is not switched off. Instead, it is literally carried away from the kinetochore down the microtubules by a dynein motor. That's the walking guy. This is really what it looks like. It has long legs so it can avoid obstacles and step over the kinesins, molecular motors that walk in the opposite direction. Personally, I'm astounded by these tiny molecular machines, how they're able to routinely and faithfully execute their functions billions of times over inside your body at this exact instant. I'm also amazed by the scientists who were able to work out how this happens in such detail that we could create realistic depictions of them like you saw in the animations in this video. But perhaps the most amazing thing is just how much is left to be discovered, like figuring out how exactly the chromatids are pulled to opposite ends of the cell. There is still so much that we don't quite know. You know, what I find exciting is that in science fiction, for decades, we've been writing about tiny nanobots that will be injected into our bloodstreams that can heal us. But what this suggests, the existence of these tiny molecular machines inside us, it suggests that there isn't a physical limit that would prevent that. And so I think it's pretty likely that in future, we will be able to develop our own tiny molecular machines that will be able to repair our bodies better than they can repair themselves.
Well, Harper, we're going to quit right here for a bit, just so that this letter is not too long. But we'll pick up right here in the next letter, and I'll show you how Darwin's theory absolutely does break down. Okay, talk to you soon.